Good morning and welcome to the very first presentation in the Biogeography and Systematics series. This is presented by the Packer Lab and the Center for Bee Ecology, Evolution and Conservation at York University. Before we begin, I'd like to let everyone know that we will be recording this talk. So if you need to head out early or if you have technical difficulties, you will be able to find the recording on our YouTube channel. Please subscribe so you will receive notifications of all of our future posts. Once the recording is available, we'll also email a link to everyone who is registered for this webinar. And just a few housekeeping items before we get started. After the presentation, we will have a Q&A period. Please feel free to use the Q&A feature, and that's at the bottom of your screen. You can pop your question in there at any point, and we'll get to them at the end of the presentation. So even though we're coming together virtually from different regions across the planet, I would like to acknowledge that I'm coming to you from the traditional territory of the Anishinaabe people, the Haudenosaunee, and the Ojibwe nations. York University also recognizes that many Indigenous nations have longstanding relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon, Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you'd like to find out more about the Indigenous cultures and treaties of your region, please visit www.native-land.ca. And now I'd like to welcome Dr. Lawrence Packer, Distinguished Research Professor at York University.
Thank you very much, Carolyn. So good morning, good afternoon, good evening to everybody. We have almost 340 people registered for this talk. From almost 40 countries and in approximately 17 time zones. So this is the first talk in the series. We've announced the first 4, but I'm happy to say we'll soon be able to announce all 11 for the year 2022. We won't have a 12th one because I'm not sure many people would want to attend a talk on December the 28th. Okay, so with no further ado, I'll introduce today's speakers. Nyaton Kitnya is pursuing her PhD on comparative study of giant honeybees in India from Rajiv Gandhi University, Arunashal, Pradesh, in collaboration with the National Center for Biological Sciences, Bangalore, India. She completed post-graduation studies in zoology with specialization in molecular endocrinology and reproductive biology. Soon after graduation, she was involved in a project on the Himalayan giant honeybees, the gorgeous Apis laboriosa, and that was her turning point to move from mice models to bees. From then on, she has been working on giant honeybees with respect to their distribution and diversity. Gard Otis was lured away from butterflies in 1975 when he began his studies on the swarming biology of Africanized honeybees in South America for his PhD degree. He was hired by the University of Guelph in 1982, where he has had a long career in both basic and applied research on honeybees. He and his associates have contributed extensively to our understanding of honeybee ecology and behavior, most recently with 2 remarkable studies of the responses of Asian hive bees to attacks by giant hornets. Gard got his first taste of the diversity of honeybees in Asia in 1986 when he observed a huge nest of Apis dorsada in Malaysia. From that experience, he felt compelled to continue research on the honeybees of Asia. During his first major research trip in 1989, he obtained specimens, photographs, and data that contributed to the confirmation of 2 additional taxa as species. His ongoing research focus is to further document the diversity of honeybees across Asia. So I now pass the screen over to Gard Otis and Nyaton Kitnya. Thank you very much, all of you, for attending, especially to today's 2 speakers.
Thanks for that introduction, Lawrence. It's my pleasure to be here today with Nyaton, my colleague from India. to share with you a bit of what we've learned about the giant honeybees over the last few years. I'd like to recognize specifically the collaborators that we work with in India that are listed there. And I would also like to just acknowledge briefly that a lot of what we're talking about would not be possible without the collaboration and sharing of specimens from a number of people. I can't mention them all, but I'll specifically mention Xin Zhao of China, Natapat Ward of Thailand, Deb Smith in the USA, Pham Hong Tai and Hank Duc Pham of Vietnam, and also Cornell University, who lent us a nice collection of Apis laboriosa drones and workers to study. So with that, let's get right into it. So I think probably everybody who's involved in this presentation today is aware that traditionally there have been 4 species of honeybees widely recognized and accepted for a long period of time. Apis mellifera, which is the bee that's native to Europe and Africa, which is the driver of honey production worldwide. And then in Asia, we have 3 species that were recognized: Apis cerana, the cavity nesting honeybee; Apis florea, the dwarf honeybee; and Apis dorsata, the giant honeybee. And at the right, you can see a simplified phylogeny of these 4 taxa, with the giant honeybees, subgenus Megapis, being the sister group to the cavity nesting bees. So here are our players for the day, the giant honeybees. There are 4 taxa that are recognized. You see in the pictures there. And you can see they look a bit different, although these 2 at the bottom here are quite similar. Apis laboriosa here, that's going to be a focus of a lot of our talk today, is a large, dark harrier bee that nests from the mountains from northern India across over into northern Vietnam. And there'll be another more detailed slide of that in a second. Apis dorsata, per se, is the widespread giant honeybee over much of Asia, going all the way from Pakistan across into Timor in the eastern part of Indonesia. And so it's basically the mainland Asian form. And then we have 2 island forms. We have Apis breviligula of the Philippines and Apis binghamai of Sulawesi in Indonesia. And during the talk, we're going to be using the names, we're going to be presenting these as species names for simplification. But of course, a large part of the purpose of the talk is to evaluate what we know about them and evaluate their species status. And these 3 taxa here are being referred to as the Apis dorsata complex. Dorsata, breviligula, and binghamai are clearly much more similar to each other than Apis laboriosa is there on the right. So I'm not a taxonomist, neither is Nyaton. So we have to start with a little disclaimer in that we're going in over our heads here and talking about things, presenting information as non-taxonomists trying to deal with a taxonomic issue. If we say anything that's incorrect or grossly laughable, please give us a little benefit of the doubt there and just let it go, or share with us later what we did wrong, because we want to learn from our experience here. But here are those 4 taxa. Apis dorsata was the first of these 4 taxa to be recognized. Apis laboriosa was described by Frederick Smith in 1871 from specimens in Yunnan, China. And as I mentioned, it's a pan-Himalayan bee. Apis binghamai was of Indonesia, of Sulawesi, was actually recognized by Smith in 1865. But at that time, he referred to it as Apis zonata. That name had already been used previously for a different form of honeybee. And so eventually then it became known as Apis binghamai by Cockrell in 1906. And I would point out then that within Apis binghamai, both Ashmead in 1904 and Cockrell in the 19-teens, I think it was 1908 and 1919, included the Philippine bees within Apis binghamai of Sulawesi. So they felt that those 2 taxa were the same. And finally, then the most recent, which isn't recent at all, but the most recent change was that Ma in 1953 designated Apis breviligula as a distinct species, the form that occurs in the Philippines. If you're interested in the taxonomy of honeybees and the nomenclature, please go to see Mike Engel's paper. And oh my goodness, I can't remember where it is. Oh, I put it right there, Journal of Hymenoptera Research, because he's reviewed the like 175 names or something that have been applied to honeybees. And I won't try to repeat all that here. So here's where we're going today. First, we're going to really compare Apis laboriosa and look at it compared to Apis dorsata, primarily of mainland Asia, but the dorsata complex as well. Looking at geographic distribution, where the bees occur in partial sympatry in parts of their range. We'll be looking at some evidence that mating flight times of drones differ. We'll be reviewing the morphological evidence that separates dorsata from laboriosa, or dorsata complex from laboriosa. And we'll also be looking at some of Nyaton's recent data on from sites where she was able to collect both laboriosa and dorsata sympatrically and compare them both morphometrically and genetically. And then after we've shown you why we are quite convinced that Apis laboriosa is a species, then we'll move on to that dorsata complex, those three other taxa, and look at some of their morphology and genetics and try to come to a conclusion of how many species we might reasonably accept at the end of this day. So Apis laboriosa, as I mentioned, Frederick Smith first described it from Yunnan, China. My sense is there were very few people exploring the mountains of Asia for a long period of time and collecting insects there, because his description and his specimens seem to have gone unnoticed by most people who have been involved in honeybee taxonomy, until finally in Ma in 1953 resurrected it as a species within the subgenus Megapis. Ma, those of you who are familiar with Ma's work, Ma was the ultimate of the splitters in taxonomy. He went from the 4 species that were largely recognized at the time he was working to a total, I think, of 24 species. And he split the Apis dorsata complex into these 4 taxa that I just mentioned and called them all species. He presented a taxonomic key to identify them. So a lot of his work was discredited because he had split so heavily Apis serrana and Apis mellifera. But nevertheless, these 4 taxa of the giant honeybees can be distinguished to some extent, and that's where we're looking today. Apis laboriosa really is understudied, and probably because it nests typically in cliffs and inaccessible locations high in the mountains. Bsakagami 40 years ago was the last person to really look in detail at the morphology of these bees. And he actually concluded that laboriosa deserves species status. He actually said that the differences between laboriosa and dorsata, in his opinion, were even greater than the differences between serrana and mellifera. But there hasn't been any other detailed morphological study since then. We do know quite a bit about its biology from the work that Ben Underwood did, and that was a third of a century ago, where he got a lot of information about the nesting biology and the biology of the bees from Nepal. But despite Bsakagami and his associates concluding that Apis laboriosa was in fact a valid species, both Rutner in his major reviews of bee taxonomy and Engel in '99 mentioned laboriosa, reviewed it, but then came to the conclusion that the evidence just still wasn't there to accept it as a valid species. In contrast to that, Michener in his first version of the Bees of the World in 1990 did recognize laboriosa among the 7 species he recognized in that book. So here's the bee. Bsakagami called it the world's largest honeybee. It is big. It's a spectacular bee with these lovely golden hairs on the thorax and the black abdomen with white abdominal stripes, which you'll see in some of the other slides. You can just see one stripe there. They typically nest, as I mentioned, in these inaccessible cliffs. This is the furthest south in mainland Asia site in west central Myanmar, with a large aggregation of laboriosa nests on this cliff. They build single combs, as do all the giant honeybees. But as you'll see, some of them nest solitarily and some nest in these aggregations. So if you were to just have specimens from mainland Asia of giant honeybees, you could instantly separate Dorsada from Laboriosa. You can see here they look very different. Dorsada is bigger, but hairier, blacker abdomen, but those are all traits that one would expect of a high elevation bee, adaptations to a colder climate. In addition, they have these golden hairs on the thorax, as I mentioned, whereas members of the Dorsada complex have black hairs. But you can instantly see that these look very different. And in fact, you can even, through binoculars from a distance, you can look at a colony and very readily tell that these Dorsada in mainland Asia always has some orange or yellow on its abdomen. The older, darker foragers here, you can see, have an orange anterior part of the abdomen and a black posterior portion. The younger bees are much paler yellow. Laboriosa over here, totally different. You can see many of these bees with the white bands clearly visible on the abdomen, on the black abdomen, and the golden hairs of the thorax. So the first thing we did in our studies was we decided to reevaluate the distribution of Apis laboriosa. We felt that there were many more records that could be incorporated from the previous assessment of its distribution. And here you see it. Here's you see what we came up with from northern India, as I mentioned, all the way across the mountains over to northern Vietnam. Just this last summer, honey hunters reported to my colleague Hank Duc Pham a couple of aggregations in extreme northern Vietnam, very close to the Chinese border, which extends the distribution to the east considerably from where it was last assessed when we did it the year before. And in addition here, we extended the distribution here to the northwest. We also got quite a few records coming down through eastern India, down, as I mentioned, that photo you had of the nest aggregation here in Myanmar. I'll point out two different clusters. This little cluster of sites up here in Sichuan, China, that looks like an outlier, and then this question mark up here in the upper right. And I point those out because if you look at a map of precipitation for this region of Asia, you can see that from northern India here, from where we know Apis laboriosa occurs, from here in northern India, all the way down across over into northern Vietnam here, all of that region is showing in medium blue, which indicates at least 150 centimeters, 1,500 millimeters of rain a year. And you can see that that rainfall pattern extends all the way up here into northeastern Pakistan and adjoining Jammu in India. We have several pieces of evidence that suggest that laboriosa does occur up here, but so far we haven't been able to verify that. And my colleagues who've looked for it in Pakistan were unsuccessful last summer. One of the things that many honeybee biologists who've gone to Asia have done when in looking at these new taxa of honeybees that have been discovered in recent years is they ask the question, well, do the drones of these different forms take their mating flights at different times of day? And if they do, this was used as one of the pieces of evidence to support species status for some of these populations. So there's two key things to see from this diagram. This is a figure I presented over 20 years ago, and there's more data that's been acquired since then. But first of all, the different patterns that are on the slide indicate the different species of honeybees. So for example, this crosshatch pattern that you see at the bottom for Japan represents Serrana. In the absence of any other species, it takes its mating flights from about 1 to 5 in the afternoon over a span of a 4-hour period. Where Serrana occurs with other species of bees, such as here in the Malay Peninsula, they fly about the same time, but for a shorter period of time. In Borneo, they fly earlier. In Sri Lanka, they fly later. So although you see generally non-overlapping distributions of mating flight times for the different species, the mating flight times for any one species are not fixed in time. They can vary quite readily from place to place. That being said, at the right here, you can see Dorsada, and the little yellow stars represent sunset at each of these locations. And in every location where they've been observed and studied, you can see that the Dorsada males take their mating flight starting just before, at, or after sunset, and they're often returning to the nest. It's so late in the evening that it's so dark you can't see them returning to the nest. I remember observing this in Malaysia the first time, and all we could hear was the loud hum of the drones as they came back to the colonies in the near darkness. It was too dark to see them. I should point out that Sulawesi here represents Apis binghamii. We also, there are observations from, name's going to escape me, from the Philippines for Breviligula that also indicates the drones are taking their mating flights at sunset or afterwards. And there's data from southern India and southern Vietnam for Dorsada. So Dorsada seems to be very, very consistent at always taking its mating flight at the end of the day. So what do we know for Laboriosa? We have data from Ben Underwood where he sat on a platform that he'd pinned to a cliff and sat about 2 meters away from a nest and recorded the numbers of drones that were returning to that nest. He has data because of rainfall. He only was able to get data on 1 day from one colony. But there you see the data. They started flying at about 12:15 and they'd finished their flight for the afternoon at 2:20. So about a 2-hour period early in the afternoon. So what do we know from elsewhere? Well, until this past year, we knew nothing. These bees usually nest in very inaccessible sites, but we were fortunate, my colleagues in Vietnam were fortunate to have 2 locations in extreme northern Vietnam reported to them, where there were moderate aggregations of about 20 colonies, like you see in this photo, that are really not all that high up, and they're not all that inaccessible. And they were able to get video cameras and set them up and video record individual colonies that they then could go through the recordings and actually observe and count the drones returning to the colonies in those videos. And this left slide is interesting because in northern Vietnam, in many sites in northern Vietnam, Laboriosa that you see here with the orange and black abdomen, and Laboriosa with its black abdomen with white bands coexist in the same sites. Here they're collecting salted water from the ground. And of interest, here's a little Apis cerana over here at the left, a very dark form of Apis cerana that for size comparison to show how big they are relative to these giant honeybees. So they were able to collect these data for Apis laboriosa drones and what they get. So again, here's Ben Underwood's data on the left. The same graph, but scaled so it shows they start at about 12:15 and they ended about 2:20. And then what did my colleagues in Vietnam get? Well, the very first drone in the recordings was observed about noon. And there were actually, I can hardly see them in here, but there were a few drones all scattered through early on, but they didn't really start to pick up in numbers till 3 or 3:30 in the afternoon. The peak of the flights were occurring from 4:30 to 6. And then very quickly they tapered off after 6 o'clock. Sunset at that location is at 6:45 p.m., which is around the time Dorsada drones are presumed to start flying. There is the red band here represents the zone of the period of time with no drones flying from either species. These data suggest that on the basis of drone mating flight times, these 2 taxa are reproductively isolated from each other. So another thing that honeybee taxonomists do, and they go to Asia, we find a new form of bee, we go, is this a species or what are we looking at here? We get some males, we get them to evert their reproductive organs, and we examine them. So male honeybees have relatively small and undifferentiated external genitalia, but they have this phenomenally huge and convoluted internal endophallus that's everted into the queen at the time of mating. And for the 4 traditional species of honeybees pictured here, you can see that the form of the male endophallus is quite different between them. Here are the two cavity nesting honeybee species. Yes, they're definitely more similar to each other than, of course, these other species over here to the right. But nevertheless, you can see substantial differences, this bump here, the length of this fimbriate lobe here, and so forth. So people, of course, when they find a new form of bee, in addition to looking at mating flight times, they say, what do the male genitalia look like? So here's what we know for Dorsada Laboriosa, and unfortunately, it's not conclusive. Simpson was the first to depict the everted male Endophallus. And it's this strange convoluted structure you see here. Gudrun Kerniger and her associates repictured it here. There are differences, but not striking. You can see that these lobes here that were pictured like that are these long lobes here pictured like that. And we can see that the other, the dorsal cornua, there are two lobes on that, two lobes over here. They're similar, but the pictures look somewhat different. This is what Underwood and his associate McKeevy depicted for Apis laboriosa. Now they argued that this wasn't different from Dorsada, but nevertheless, if you look at the ventral cornua here, you can see this long segment at the base and the branch occurring distally, which looks very, very different from what I see over here in Dorsada. So right now it looks like it's different. They argued it wasn't different. And Gudrun Kerniger and associates argued that we really need to look at it again because it's not very conclusive. So those of you out there listening, if you have access to Laboriosa drones, here's a project. So as I mentioned at the beginning, our collaborators who've shared specimens with us, we've been able to compile a fairly large data set of samples to examine, both workers and drones from across Asia, across their ranges in Asia. We don't have huge numbers of bees here, Breviligula and Binghamai. We don't have huge numbers of drones to look at. Yes, there are small numbers in some of these, but nevertheless, we have a wide distribution of bees, which I'll just point out when Bsakagami published his paper and looked at Laboriosa versus Dorsada, almost all of his Laboriosa came from Nepal, the vast proportion of them. We have a much wider range here in our specimens to examine. And at this point, I'm going to turn this over to Nyaton, and she's going to pick it up from here to talk about some of her work evaluating the morphology and genetics of these bees.
Thank you, Gard. Thank you so much. So as Gard had walked us through the geographical distribution and the behavior evidence, now let us move to the morphological evidences. As Gard mentioned in the just previous slide to this, for morphological examination, we have a specimen from across Asia. However, here I would like to point out one point that for the characters that require a dissection, we have a small subset of the specimen that we have right now. So as we move for morphological evidence for Apis laboriosa, earlier Bsakagami and al. In their comparative study between Laboriosa and Dorsata, they reported that Laboriosa is distinct from Dorsata. There is a distinct variation in the Ocelot position, as you can see from these two pictures. In Apis dorsata, the Ocelot is positioned in a much raised platform. On the other hand, it is relatively low in Apis laboriosa. At the same time, the distance between the compound eye and lateral Ocelot is much wider in Apis laboriosa, unlike those in Apis dorsata complex. So this particular character, we were interested to examine across the 4 giant honeybees that we have. Then this particular character consistently separates the Laboriosa from the Dorsata complex. Next. I'm sorry, so Gard will be helping me to move the slides. And if we look at the head of the worker bees of these 4 groups of the giant honeybees, as you can see from this picture, Laboriosa has a wider space between the lateral ocella and the compound eye, and it is relatively low in the other 3 giant honeybees in Dorsata, Breviligula, and Binghamai. Next. So if we look these pictures carefully, as you can see from these pictures, Apis laboriosa, the compound eye in Apis laboriosa is relatively narrower. On the other hand, the head in the Dorsata complex is narrower. So we were interested in looking at this particular character, that is exterior eye to eye width and interior eye to eye width. And when we measure this character across these 4 giant honeybees, Next. This is the result that we got, and from this figure we can see the proportion of this these two characters. Laboriosum is distinctly separated out from the three giant honeybees, and these characters seem to separate out Dorsada from the other two Taxa. The letters in this particular graph represents the significant differences, and thank you. So, and another character that here I just wanted to mention the table represents the mean SD and the sample size that we examine. Currently we examine the lesser number of sample size for this particular character, but we look into more specimens. Yeah, next. Another character that we look into is the Miller space. Miller space is the area between the lower edge of the compound eye and the mandible. Again, in Bsakagami et al. In their study reported that the Miller lens separate out Laboriosa from Dorsata. So as you can see from this picture, Dorsata has a shorter Miller lens and Laboriosa has a longer Miller lens. Again, we look this particular character across the 4 giant honeybees, and this is the result that we got. And as you can see from this figure, this Miller lens distinctly separates out Laboriosa from the other 3 giant honeybees. And this particular character, the Miller lens, does not separate out the other 3 giant honeybees, that is Dorsata, Bingami, and Breviligula. At the same time, the Miller width does not separate the 4 giant honeybees, and this character does not seem to be a good character to separate to distinguish the 4 giant honeybees. Also, the tables represents the sample size and the mean NSD and the SD value and the range. Next. So as we move on from head to the abdomen segments, that is the sternites, this particular picture represents the sternum 2. The area shown in the show indicated with the arrow, that is glandulus. Glandulus in the Apis laboriosa is weakly curved, unlike those in the Dorsata complex. Next. Again, in sternum 3, the glandulus in the sternum 3 at the midpoint is curved posteriorly. On the other hand, anteriorly, sorry. And on the other hand, in Dorsata complex is curved posteriorly. So this is also one of the characters that distinguish Laboriosa from the other 3 giant honeybees. Next. Another character that we look at is the spherical plate. Spherical plate in Apis laboriosa is relatively wider, and the posterior edge of the spherical plate is distinctly different from the other 3 groups of the giant honeybees. So these are the different characters that we observe in the worker giant honeybees that separates laboriosa from the Dorsata groups. The next, then we move on from worker bees to the drone bees. If we examine the drones of the 4 giant honeybees, this is one of the characters that distinctly separates laboriosa from the 3 groups, that is the ocelli structure. As you can see from this picture, the ocelli in the Dorsata group is placed on a much strongly raised platform. It is much projected and is close together. On the other hand, the ocelli on Apis laboriosa drone is widely spaced, and it is positioned on relatively flat platform. If we again look this particular character from this angle, we can clearly see in case of Apis laboriosa, it is barely visible and it does not extend beyond the plane of the compound eye. On the other hand, the ocelli in the 3 groups of the giant honeybees, it is prominently protruded. So based on these morphological characters, laboriosa meets the criteria of valid species. Next. And this is one of the picture where we can see the ocelli, the structure and the position of ocelli in Dorsata complex is largely dome ocelli on very obviously pedestal, and it is absent in Apis laboriosa. So these are the different morphological traits that we observe in the 4 groups of the giant honeybees. And this shows that Apis laboriosa seems to be different from the other 3. Another approach that we were interested in is the comparative study of Laboriosa and Dorsata from the sites of sympatry. In our study, we observed 5 sites of sympatry in Arunachal Pradesh in Northeast India. That is, the sites of sympatry are marked in a color, a different color, that is blue and orange and green. And along with the specimens for our analysis, we also included specimens from where we could collect only Apis laboriosa and from where we could collect only Apis dorsata. The graph on the right-hand side shows the elevation and distribution of Laboriosa and Dorsata in Arunachal Pradesh. These are from the data that we collected. Laboriosa is in a green and Dorsata is in a blue. So this shows these two giant honeybee, at least partially, shows ecological and altitudinal overlap. Next. So we wanted to look at one line of evidence is the traditional morphometric analysis. For traditional morphometric analysis, we use the 17 characters, that is the malar length and malar width. The ocelli structure, that is the distance between lateral ocelli and compound, the interocular distance, and also the indica when in the hind wing and different wing venation angle in forewing. So these are the different characters that we use. And based on the measurement of these 17 characters, we got the data, and on those data, we ran a cluster analysis, and the dendrogram shows Laboriosa and Dorsata forms in a separate clades. As you can see from this figure, in this figure, the color code represents same as those used in the map, except for Bangalore specimens, which we didn't show in the map, but here we code in a pink color. As you can see from this figure, the green color, the specimen, the Dorsata from Tudnu and Kalapahar are clearly separated out from Laboriosa from Tudnu and Kalapahar, and the same pattern can be seen from the Nag Mandir, that is in the blue color. The Laboriosa collected from Nag Mandir are clearly separated out from Dorsata collected from Nag Mandir. Next. So with the same data set, we ran a principal component analysis, and this is the figure what we got. Here in PC also, we can see these two giant honeybees are separated out into 2 distinct groups. The solid dots in this particular figure represents the specimens from the sites of sympatry, and the open dots represents the specimen from sites of allopoetry. Okay, and some of the characters that are responsible for separation of these 2 clusters are the ocelli structure, the malar size, and wing venation angles such as G18, O26, and N23. So from morphometric analysis, we can see these 2 are 2 distinct groups. Next. On the same set of specimen that we analyzed, we use it for morphometric analysis. We conducted a genetic analysis. Based on CO1, we can see Dorsata and Laboriosa forms a separate clade in spite of their co-occurrence. Instead, Apis dorsata from Northeast India forms a clade with Apis dorsata from Thailand. And at the same time, Apis dorsata from Northeast India forms a clade with Apis laboriosa from Northeast India forms a clade with Apis laboriosa from Nepal. Interestingly, the South India dorsata separates out from Apis dorsata of Northeast India, and this agrees with the data from Deborah Smith and team in their study. So based on our analysis on the specimens collected from sites of sympatry, and when we look at the 2 lines of evidences, that is the morphometric and the genetic evidences, it also supports that the Laboriosa is a distinct species. So here I end my presentation. I would like to switch next to Gard.
Thanks, Nyaton. So I think from what we've presented so far, I suspect you're all in agreement now that Laboriosa is a distinct species from the members of the Dorsada complex. But what about the Dorsada complex itself? We have these three different forms: mainland Asia, Philippines, and Sulawesi, Indonesia. As I mentioned before, it's easy to identify Dorsada with the orange or yellow on the abdomen, as opposed to these black abdomens with white bands. But generally, it's not real advisable to define a species based on just color differences. That's relatively minor. So what about other traits? So one of the characteristics, I guess you could say, that has been suggested as a means to recognize at least Breviligula as being a different species from Dorsada is the belief that Dorsada typically nests in these large aggregations. There's a famous tree a bit east of Bangalore in India that's, I think, has over 300 different nests on it that's been suggested to be put forward as a UNESCO World Heritage Site. I haven't seen that tree, I'd like to see it. But so they frequently nest in large aggregations on emergent trees or on water towers, on buildings and so forth. That being said, it's not unusual to find solitary nests. That's just the one you see on the right. That's also Apis dorsata. These island populations, Breviligula and Binghamai, typically nest solitarily, but on one occasion, as I were headed to our study site in Sulawesi, I saw we passed a tree that had 3 Binghamai nests in the same tree. So they too occasionally will aggregate their nests. I don't think this is like a character that can really be used to differentiate these taxa. I just throw this slide in again that Nyaton presented because when we compare the interior eye-to-eye width and divide it by the exterior eye-to-eye width, you can see here, again, our sample size is small. We need to beef this up. But Laboriosa clearly looks like it has a much larger ratio than the other 3 taxa. So far, at least, Dorsada here looks like it may be separated from Breviligula and Binghamai at the bottom. Statistically, they are, but we need a larger sample set because as we get more samples, this range is going to increase on each of these. And whether it'll remain non-overlapping like you see here, or whether it will begin to form overlaps, I don't know. If there form overlaps, of course, it's not a great character for basing a species designation on. But on the basis of this right now, and also the basis of color differences, I would argue that Dorsada looks to be like it could be recognized as being distinct from the other 2 island populations. So I show this picture because, as you've seen, Breviligula and Binghamai are more similar to each other. Their head shapes are more similar. Their color is almost identical. Ma, when he recognized Breviligula here at the top as a distinct species, based his description primarily on these 4 characteristics. And we're going to go through each of those. So his first characteristic was the less strongly raised ocelli in Breviligula. That is evident in this picture. You can see here the ocelli they're raised, but they're not raised nearly as much and domed nearly as much as they are in Binghamai. We have not examined this yet in our specimens to see how consistent this is. But I would take that as a relatively minor character. I don't know that I would want to base a species on a trait like that. I could see recognizing them as subspecies, for example, but perhaps not species. And then we have these other 4 traits: the malar area, malar length and width, the abdominal sternite 5, and the tongue length, the glossa. So when we look at malar length and compare the taxa, again, we saw the slide before, Laboriosa has a much longer malar space. The other 3 taxa don't differ, so that's not helpful. And Breviligula and Binghamai do not differ from each other, unlike what Ma reported in his species description. Ma reported that for sternum 5, that Breviligula is relatively shorter and broader, so a bit more rectangular in shape compared to the more square shape here of Binghamai. So again, we haven't looked at a lot of specimens, but Nyaton has dissected a few and measured this. And so here's what we get. Yes, sternum 5 is statistically longer in bingami, shorter in Breviligula. But you can see here that even with just the small number of samples, 5 in each case, that their ranges almost overlap there. And as we measure more specimens, for sure that range is going to increase a bit, and these are for sure going to overlap. For sternum 5, the width, they already overlap extensively, so that's not terribly informative. So we took a ratio of these to see if the ratio would differ. So when we divide the width by the length, so I've got this wrong up here, this should be W versus L. You can see that as at least for our small sample size of 5 and 5 specimens each, that these distributions do not overlap. This may be a defining character that would allow us to separate as species Breviligula versus bingami. But I think I'm pretty suspicious that as we get more data and more observations, that these ranges will expand, and there's a good chance they will overlap. Finally, the tongue length. So Apis breviligula, it is the short-tongued honeybee. That's what it means when you translate the Latin. Nyaton was able to relax and extend some of the tongues. She had a bit of difficulty because in many cases the glossa wouldn't fully extend. But for those specimens where she did get what she felt was a full extension of the tongue, there you see our short-tongued bee from the Philippines and our long-tongued bee from Sulawesi. And if you compare tongue lengths, the total tongue lengths here, it's again small sample sizes, but the data we have right now for Breviligula, the short-tongued bee, it seems to be almost identical to Apis bingami, and no difference there whatsoever. This does not seem to be a short-tongued bee in particular, and it doesn't seem that that character is going to hold up. As you might expect, the Laboriosa is a bigger bee. It has a slightly larger tongue length, a longer tongue. And Dorsada tends to have a shorter tongue. This is an interesting little population from the Andaman Islands in the Andaman Sea. And they're smaller bees and they have a much shorter tongue. This would be Dorsada, maybe a subspecies of Dorsada, I don't know, but it's Dorsada. So it doesn't look like tongue length's a good character either to differentiate Breviligula, well, any of these, with any of the taxa within the Dorsada complex. And finally, we turn to genetic relationships. There have only been 2 analyses so far that have included all 3 taxa within the Dorsada complex, Breviligula, bingami, and Dorsada. Lowe et al. Have Breviligula as being the sister group to the other 2. In Deb Smith's analysis, she has Breviligula as the sister group to the other 2. What is somewhat unsatisfying in these 2 analyses is that Lowe et al. Have Laboriosa here embedded within the Dorsada complex. And from everything we've presented here today, that clearly is not going to hold up. Deb, on the other hand, Deb Smith, did not have specimens of Laboriosa in their analysis, and she and her colleagues inferred the position of Laboriosa based on some previous genetic studies and also by the morphology. So nevertheless, Breviligula separates out in both analyses on a separate clade from bingami, on a separate clade from Dorsada. So if we want to accept genetic differences within a clade, genetic species concept, then you could perhaps accept all 3 of these members of the Dorsada complex as being valid species. So there's our data. There's what we have so far. You can tell that we still need to beef up some of the sample sizes, and it's a work in progress. But the conclusions at this point, so if we compare Laboriosa and Dorsada, I don't think there's any question Laboriosa is a species. We've got mating flight times that in both cases, even though Nepal and Vietnam, they fly, the Laboriosa flies at different times of day, they still don't fly close to sunset. So they seem to be reproductively isolated. It looks like the male Endophallus may differ, but that needs to be verified. We have several sites where they Dorsada and Laboriosa co-occur, and yet in India, from Nyaton's analysis data she presented, there's no evidence of interbreeding yet from, or interbreeding from either the morphometric or the genetic studies. Laboriosa workers have several odd apomorphies that would distinguish them from Dorsada. The ocellar characters, this ratio of the interior eye width to the exterior eye width, the pictures she showed of the worker sternites 2 and 3 in the Spherical Plate. And in the drones, their ocelli are like night and day differences. Anybody could pick those up and look at them and tell they're different. And I forgot to put in here another odd apomorphy is the longer malar space in Laboriosa compared to the other taxa. And all the recent phylogenies that have been done all separate Laboriosa out as a separate clade, distinct from the others. The position of that clade may vary a bit, but nevertheless, it's a distinct clade. So all the evidence suggests that Laboriosa is a valid species. Where we run into more problems is within the Dorsada complex. First of all, they have allopatric distributions with Breviligula and Binghamai being on different clusters of islands separate from the mainland. They all take their mating flights at the end of the day at or after sunset. Their nesting strategy differs a bit, but not diagnostically. Dorsada differs in color and in those head proportions that we mentioned. I get the impression from looking at specimens that Dorsada actually has a different head shape from the Breviligula and Binghamai as a group. The ocelli of Binghamai are slightly more raised than in Breviligula. The sternum thigh proportions look like they're a little bit different, but with more data that won't probably hold up. Genetic analyses separate all these taxa into separate clades, but the positions of those clades. Sorry, it says they're not congruent, but within the Dorsada complex, they're relatively congruent. And so in a sense, there's evidence that you could take to say that all 3 of these deserve species status. I know you probably are going to want us to come down and off the fence and make a definitive statement. And I realized I added a slide yesterday at the last minute. I thought we can't leave everybody without telling you what we think at the end of the day. So Dorsada, Laboriosa, clearly distinct species. And then I would argue that the island populations here from the Philippines and Sulawesi are similar enough that they should be considered subspecies, not full species. And if they were subspecies, they probably, I'm not a taxonomist, but my sense is they would fall under the name of Apis binghamai, since it was described much further back in time than Breviligula was by Ma in 53. So they would have probably those names. There should be more study of these, but I think we're going to find that as we examine these more closely, the kinds of differences we're going to find are going to be relatively small. And I'm not sure they're the kind of differences I would want to base a species description on. So that's where we sit. And in the end, then, I just would like to state some of the people I'd like to acknowledge. I've been helped so many times by so many people, scientists, villagers, honey hunters, who shared their homes, they've shared their knowledge, they've shared their time with me, and I can't thank everybody enough for all the help I've gotten over the years. A special shout out to Mahadir Mardan, who I first met as a grad student here, studying giant honeybees at the University of Guelph. And I often think of him as my brother from another mother. He's my Malaysian brother, and we share so much in common. And finally, as a grad student and then later in life, I became pretty close to Dr. Michener, who, for many of us who knew him, he was the apodologist without par, and he mentored many of us. And I'll turn it over to Nyaton to acknowledge the people she'd like to speak to.
Thank you, Gard. So I also would like to acknowledge my colleagues and friends who have helped me throughout the project, but help us in different areas. And also not forgetting the locals who have been supporting us during our field trips and sample collections, particularly in Northeast India, Uttarakhand, and Andaman Islands. And also I would like to acknowledge the Department of Biotechnology India, University Grant Commission India, NCBS for funding, and also NCBS and Rajiv Gandhi University for the facility where I could able to carry out all of this work. So thank you so much, everyone, for being here and listening, talking us about what we have been doing, and we'll be happy to take any questions and any discussions. Thank you.
Fantastic. Thank you very much. That was wonderful, and I'm convinced. So I'm going to go to the questions that have been put, and I'll deal with them in sequence. Hello, Wanja. Have these bees ever been put in a hive for honey production?
Nyaton, do you want to answer that?
Sorry.
I'll answer it. I'll answer it. All the giant honeybees nest in exposed situations, building a large nest. My understanding is they need to have access to see the sky to orient their foraging dances. So I don't think it would be successful to put them in a hive. And it really wouldn't be very manageable because the hive would have to be a meter long by 3/4 of a meter tall and a single comb wide. It would just be an unworkable situation.
Okay, thanks. Okay, so from Eugene Scapula, what is the large peak that seems to depict a Dorsada drone mating flights around noon in Borneo?
Oh, I'll go to that. I pulled up this view so you can see these. This one? I presume you mean this one. That was the data for Apis luensis. That is a highland form in Borneo that co-occurs with both Apis koshevnikovi and Serena from elevations of about 1,600 to 1,800 meters. And then from there up to nearly 2,500, 2,600 meters, Apis luensis occurs in the highland. And I'll just point out something about honeybee taxonomy. Apis luensis was described in, I can't remember the years, about 24 years ago. But the species description is such that you couldn't pick up a bee on a pin and hold it in front of you and look at it and identify it. And because of that, and they're very, very similar in most respects to a high elevation, dark black form of Apis serena. So depending on your species concept, the people who, the male Endophallus doesn't differ from serena. Some of the more conservative people like Michael Engel would say that that represents a subspecies of Apis serena. He does not acknowledge it as a species, whereas given its habitat, its elevation, the fact it's very distinct, the fact you can find them for Apis luensis, koshevnikovi, and serena all foraging together on the same flowers suggests to me that it's a distinct species, but there needs to be more work done there. That's what that is.
Okay, thank you. So Zachary Wang has said that he's heard Laboriosa is also in Lingji, Tibet, and also Nyingchi, Tibet, 400 kilometers east of Lhasa.
You need to send me those data, Zach. We'd love to see it. This is what we compiled in 2020, everything we could find written and published. And we searched and searched and searched to try to pull up records. And I'm sure it's incomplete. I'm sure there's some other locations, but there is a site here, and there are sets here in Tibet that are included in this distribution map.
Okay, thanks. So I'm going to apologize if I'm mispronouncing anybody's names. I should do that at the outset, really. From Babu Saddam, Laboriosa is found in Uttarakhand, India also. These are comments rather than questions.
Sorry. Can you repeat that? Found where?
I'm Uttarakhand. Uttarakhand.
Uttarakhand is right here. These are all Uttarakhand specimens right here.
All right.
But we have not yet been able to confirm. It seems like it occurs right over near the border with Pakistan or the contested territory in Jammu. But so far, nobody's been able to go there and find them and collect them and verify them there, or in northeastern Pakistan nearby.
All right, thank you. From Comment Ken, I've read that Laboriosa migrates down in winter and up in summer. Does Dorsada migrate seasonally? Is Sympatry only in winter? Is there any breeding in winter?
Yeah, putting a wrench in the thing. Are you there? I'm trying to find the data here for the elevational distribution. Where was that? Oh, it's further on. Sorry. Where's our elevational thing? Here we go.
Yeah, there. Yes.
I can't get my mouse to. There we go. Yeah, so this elevational distribution is a little bit misleading because that just takes any records from any month of the year and throws them together. So this extremely low elevation record here in the dark green for Laboriosa, below 500 meters, probably represents a wintering cluster of bees. They do move up and down the valleys, both in Nepal and in Arunachal Pradesh, this region where Nyaton has worked. I can't remember if I took those slides out. I had some suggestions of what we think might be going on, but in the absence of any data, it's really speculative. But our impression is that the bees migrate up these river valleys up through Arunachal Pradesh, up toward Tibet to the north, and that they probably reach Tibet probably in July or August, and then migrate back down. But at some of these intermediate elevations, like from 500 to maybe 1,300 meters, we have evidence that probably Dorsada and Laboriosa nest in very close proximity to each other in northeastern India. At the same time of year, sorry, at the same point in time.
Okay, so I like to call Apis laboriosa the abominable honeybee because of it's found at such high altitude, and I'm hoping you'll use that as a common name.
I don't think so, but anyway, good fly, good try.
Go on. Diana Hernandez has asked, well, first of all, she says that's a really interesting talk. Is there any information on differences in microbiomes between these species?
No.
Because there are no data.
No data.
Okay, so you should get, maybe Diana would want to collaborate with you to get some. James Dory, thank you for a fantastic talk. He asks, why do you think the drones are flying after dark? Predator escape, sexual selection, or reproductive isolation? And he also asks, do queens also have modified eyes if they're also flying in low light conditions?
I've thought about this question many times, and I don't have an answer. I actually expected when we went to, for example, Sulawesi, that in the absence of some of the selective pressures that one would find on the mainland, that they might have mating flight times that would be earlier in the day. That did not prove to be true, although we don't have a lot of data from Sulawesi. We have basically just an observation. And for queens, there's boy, I don't even know of any specimens of queen laboriosa. They might have some at Cornell. It's really hard to get queens of these bees, so I don't think anyone's looked at them. So hard to get queens. You have to technically kill a whole colony and look through it to find a queen.
Jason Gibbs just sent an interesting comment on the chat. He said the abominable honeybee hasn't caught on as a common name, at least not Yeti. So Chris Darling asks, what do you mean by genetic analysis?
I was afraid you'd ask that. Sorry, I'm kind of taking over from Nyaton at questions here. Nyaton, you can talk what you did. I think that was clear.
Okay, genetic analysis. Thank you, Gard. Genetic analysis here we mean is like we only did with one of the mitochondrial gene, that is CO1. Okay, maybe you can pull out the.
No, that's enough. That's fine. You explained it. On this slide, I think this is the one you're talking about, genetic analyses. Deb Smith used SNPs, and it was nearly, she's on the line here, she could probably comment, but it was something like 960 SNPs, single nucleotide polymorphisms scattered through the genome. So it was a pretty large data set spread across all of the chromosomes. I cannot remember exactly which genes Low et al. Sequenced, but I believe they had one mitochondrial gene and 2 nuclear genes that they had DNA sequences from. And given that those 3 sets of data, I'm really surprised that Laboriosa ended up nested here within the Dorsada complex, as opposed to being like basal to the rest of the Dorsada complex.
Yeah, that'll depend on the method of analysis to some extent, and
Yeah,
I'm not a
geneticist. I'm not a geneticist, and so I'm out of my league there. That's where, as a taxonomist talking about or non-taxonomist talking about taxonomy, I get in deep water.
Okay, so thanks for that. Alison Malay asks Sajesh Vijayan says, amazing talk. Were you able to compare how closely the bees from Andaman Islands were related to the other clusters, e.g. South India and North India and Northeast Asia?
Nyaton, you'll have to answer that.
Okay, yeah, we look at the morphometric analysis of Dorsada bee from Andaman Island and from different parts of India. So Dorsada from Andaman Island seems to be much smaller in size, but yeah, that's all we could find in different characters. Overall size is very small compared to the Dorsada from mainland India. But if we look at, when we analyze it, separate out totally based as a different group. Based on the morphometric character that usually used in the study of honeybee. And for the genetic analysis, we are still analyzing it. We are processing the samples. Yeah, I think I answered what it meant. Yeah.
Okay, thank you. So Alison Malay, when hives are congregated, are they considered separate reproductive units? Will the drones mate with neighboring hives? I guess potential queens from neighboring hives.
I think I'll answer that. I meant to mention that in that slide about mating flight times. All the honeybees have a somewhat similar mating system in that males leave the nest, they fly off to somewhere. We know that there are congregations of them that we call drone congregation areas. Queens leave the nest to go off to mate. It's been inferred that mating occurs in these drone congregation areas, although no one's actually ever been able to track a queen to know exactly where she mates. But nevertheless, they leave the nest and they go off and they mate with males from many males from many different colonies. Actually, the analyses of Dorsada, when they do genetic analyses of offspring from a nest, they frequently find as many as 50, 60, 70, even 100 different males have mated with a queen. So they're flying off and mating with males from many colonies from a wide area. Not from their own, well, probably some from other nests within the aggregation, but probably also drones from other aggregations as well.
Thank you. From Babu Saddam, is this species commercially available for rearing and honey production?
Nyaton, you want to answer that?
I think you can go ahead, Gard. Yeah.
Oh, okay. No, we can't. It's all honey hunting for the giant honeybees. It all involves climbing trees or descending on ropes from cliff tops to get at Laboriosa colonies on a cliff. There's no, the closest to management comes from a few sites where people put up what are called rafters. And these are angled logs, basically, or angled planks. They often smear beeswax on them to give it an odor of the colony of the honeybees. And then migrating swarms will find these and nest on them. And in some cases nest at relatively low heights, especially for like Dorsada. So this has been documented in, in southern Vietnam, in Sulawesi in Indonesia, I think some other islands in Indonesia, Bangka Island, where people put up these rafters and get the bees. So once they're on a rafter, once they're on a plank, You can go to them, you can smoke them lightly, you can get the bees to move off the honey portion of the nest and cut away the honey, and the bees don't leave the nest. And you can actually get 2 or 3 harvests of honey, but that's the closest to management that anyone's been able to achieve with any of the giant honeybees.
Sounds pretty dangerous to me. John Gibson, when they migrate up and down the valley, are they building a whole new nest at the nest site? How time consuming is this, if so?
Nyaton.
Pardon, Lawrence, can you please repeat the question? Yeah.
It's just disappeared, but I remember it. Laboriosa flies up and down in altitude. Do they build a new nest when they change in altitude?
I think from the study from Underwood, they form during winter. If they come down, they form a winter clusters, but I don't know if they, we don't have any data, any record if they change the sites of building in different seasons.
But Underwood in Nepal, he had, at the higher elevation sites, the bees only arrived there like in late July and built a nest, built a comb, and then stayed for maybe a month and a half or 2 months and then left. So yeah, there's a huge cost there in terms of build. They also will move, fly, hang out in a bivouac, like just without a combless cluster, and then take off and fly again as they're moving. We know so little about this. And if you can figure out a good way to put markers on bees in these swarms and track them, that would be a wonderful thing to do in a mountainous region. I personally don't know how you'd do that, but we'd love to know answers to those kind of questions.
Okay, thank you. And the last question comes from Art Davis. Oops, I've just lost it. It's in the chat. And it was about the ocelli. Okay, here we are. Thanks for the very inspiring talk. Is the inference that the large raised ocelli in the Dorsada complex potentially responsible for, I can't see responsible for night flying, e.g. foraging workers, drones out on mating flights versus the mating flights of Laboriosa occurring diurnally? Moonlight important? Are there behavioral studies where ocelli have been blackened, covered on experimental bees that might suggest that blocks their ability to fly and navigate at night? All the best from Saskatoon, Saskatchewan, Canada.
All the members of the Dorsada complex, that'd be Dorsada, Bingamine, Breviligula, are all known to forage nocturnally. And we have not, there's a correlation in general that bees and wasps that are active at night, like Megalopta, the halictine. Is that Megalopta or Megaloptera? Anyway.
Megalopta.
They also have domed and large ocelli. So the domed and larger ocelli are almost certainly related. I think they're almost certainly related to detecting light and flying at night. And I think in the case of Dorsada complex versus Laboriosa drones, again, Laboriosa drones are flying during daylight, and the Dorsada complex drones are flying at the end of the day and almost pitch black by the time they're off and returning to the nest. So again, I think those prominent ocelli that are raised up and large ocelli and domed ocelli have something to do with detecting light intensity at those low lights, but low lux. But we've talked with our colleagues in India, we've talked about doing a detailed analysis of eye structure and ocellular structure. And that's sort of sitting on the burner there waiting to have the fire lit. Someday, maybe you should do it, Art. You have nothing to do out there in Saskatchewan except shovel snow. So you could probably do this in your spare time.
All righty. Well, that's it. No more questions. So I'd like to thank the speakers in particular, but I'd also like to thank all of the people who are attending this talk. From all over the world. Some of you stayed up late into the night. I think, what time is it where you are, Nyaton?
It is 10:47. It's almost going to be 11 p.m.
Okay, well, you do pretty well late at night, so maybe you should go out and catch some of these nocturnal bees now. Okay, well, thanks. That was a great talk. Lots of great questions. You've obviously inspired a lot of people. We had lots of attendees from all over the place. And well, thanks a lot. That was a lot of fun.
Well, I just like to thank everybody. And I can't imagine 3 years ago, before the pandemic, actually being able to put together a seminar that involves Nyaton in India and me here and people in however many you said, 16 time zones or something, and all being able to join in and all participate in this thing. It's amazing. And I thank you guys at York for putting this together. Thanks so much.
You're most welcome. All righty. So we're going to sign off. And as we do that, this will end up being recorded and stored on a computer somewhere to be mounted on the internet later. Caroline has put up the next 3 talks, so everybody knows what's coming. Paul Williams, when did bumblebees arrive in South America? That one will not be recorded. Catherine Odenaka and Thomas Onufurko, Phylogeny and Biogeography of Kleptoparasitic Nomadine Bees, Epiolini and Nemata, and Michael Orr, the biology and evolution of the subfamily Anthophorini Apidae. All righty, thank you all, and goodbye.